Hello everyone. On this video we are going to be seeing how this vaccine that was produced by Pzifer is also called the BNT16 2B2 vaccine. Okay, and now in order to understand the principle on which this vaccine operates, we have to start from the structure of the COVID-19 virus. So this is the structure of the COVID-19 virus. This red stuff that we see up here are known as the spike proteins. Okay, and then this helical structure inside here is actually the genome for COVID-19 virus. And then we see this red portion, okay, this red portion that has been demarcated here is actually the gene that codes for the spike protein. So what the Pzifer company did was that they came in here and read the nucleotide sequence of this gene for that codes for the spike protein and then produced the messenger RNA, okay, for that spike protein. So this red stuff here is actually the messenger RNA of the spike protein. Now, injecting this messenger RNA directly into our system will not work. The reason being that our enzymes are going to cleave and destroy this messenger RNA. So what Pzifer did is that they englobed this messenger RNA into a lipid nanoparticles. Okay, so inside here we have our messenger RNA, and this stuff around it are the lipid nanoparticles. Now, in this form, our enzymes can no longer destroy our messenger RNA. Also, due to the fact that this messenger RNA is very fragile, if we allow it at room temperature, it can easily degrade. So that is why Pzifer produced congelated boxes at less than 70 degrees Celsius in order to store this vaccine in it so that the messenger RNA will not degrade. Okay, so in our vaccine, these are the particles that you will find inside. That is the messenger RNA that has been enveloped inside lipid nanoparticles. So this vaccine or these particles are then introduced into our system. So in the system, the lipid nanoparticle layers of our vaccine is going to fuse with this lipid bilayer of our cells. When that fusion takes place, the messenger RNA will be introduced into the interior of our cells. Now, inside our cells, this messenger RNA will be translated into the spike proteins, okay? These spike proteins are immediately transported to the surfaces of our cells, okay? At the level now, found at the surfaces of our cell, a special form of immune cells known as antigen-presenting cells are going to pick this up, these spike proteins, process them, and present them to another special form of immune cells known as the T helper cells. Okay, the T helper cells. The T helper cells. These T helper cells will then activate another type of cells known as the B cells or the B lymphocyte. These B cells are the ones that will produce antibodies. Against the COVID-19 virus, due to the fact that they recognize the spike proteins that were shown to the T helper. So they will produce specific antibodies that will recognize the spike proteins on the COVID-19 virus. and therefore these antibodies can easily target the COVID-19 virus. Another thing that happens is that these B cells, some of these B cells would remain in the system as memory B cells, okay? Some of these B cells will remain in the system as memory B cells. Now, all what we have been explaining takes place as we are vaccinated. We are not yet infected with the COVID-19 virus. All this takes place when we are vaccinated. Now, when we are actually infected with the COVID-19 virus, what happens? What happens is that these memory B cells will immediately produce antibodies, okay, against our COVID-19 due to the fact that they will recognize these spike proteins. And when they produce these antibodies will immediately attack the COVID-19 virus. This will also stimulate immediately a cascade of immune responses, okay, that will result in the immune cells attacking the COVID-19 virus immediately and destroying it. This way, the COVID-19 virus can no longer infect our cells. Okay, so basically, these are the activities, or this is the principle on which the Pzifer anti-COVID vaccine operates. Okay, so thank you for following the video.